EP Biology on Friday. This is a learning day, and you're going to accomplish a big must-do just by joining the Pear Deck with me. I'm going to start in a minute and participating fully in it. So I want to see you writing things when it says to write things. I want to see you dragging those dots. I want you to see you picking answers. That's the only way I can write you off as 10 out of 10 on this, because if I don't see your name in there doing something, I can do. Okay, so please take that seriously. This isn't self-paced. We're going to do this together. I wrote this myself. I spent an awful lot of time on it to make it so that it's good diction and it's all saying things the way you need to hear them, nice and exact and precise, and yet also simple. So this would be a good idea to kind of fill in a note sheet that I made for you. It's optional, right? Participating in the Pear Deck is the main thing. I just want you to participate. But I also made just a really short kind of take notes as you go worksheet that has blank pictures of the graphs, and then you can fill them in as you see the picture come up, and that might be really helpful, because then after you do this, you're going to do a free energy graph worksheet that's exactly based on the Pear Deck notes I'm about to give you. So let me show you where that is. Taking it out of here for just a second. Let's go to the classwork tab. Here we go. There we are. Okay, so popped a few more things in here since last time I saw you. So this here, right here, that right there, the notes for Pear Deck on enzymes. It's optional, right? It's not very long, but it has the blank pictures of the graphs on it. So if you wanted to open that up and just print it out and have it, you know, to scroll on while we're talking, it might be a good idea if you like to have your stuff organized like that. Okay, and then the must do then that after that, this is what I want you to do afterwards, right? So this is the worksheet that that has you practicing those free energy diagrams that I'm going to be talking about in a minute. So you may want to print that one out too, or just have it open for yourself. Okay. All right, and then I'm going to also call attention to this one up here. Okay, so this will be another ed puzzle that's not practice. This is going to be summative. This one is your pre-lab to next week's big lab. So this will be your homework. Okay, so don't open it now. Don't do it now. We're going to do the Pear Deck now, but just so you know, that's a summative one, and you want to pay really close attention to it. There's some really interesting stuff about lactose intolerance in there. You're going to enjoy that. I found out things I didn't know, so I hope you enjoy that. Okay, so now, good. All right, we're going to go start the Pear Deck now. That's what we're going to do. So, let's see, on this in a bit. Chrome tab. Pear Deck. Start. Ooh, I have a feeling you can't see that, can you? Hold on. Hold on, I gotta fix it. I gotta fix it. Hold on. Right, let me take you to the right screen here. Okay, let's try this again. Here we go. Okay, here's the screen you need to see. Yes? There, you can see it, right? There you can. Okay. So now what I need you to do is go in there. Oh, thanks, John. All right. And we're going to do this Pear Deck together as a nice Friday learning exercise. Hopefully this will work for you. So just take a few minutes and get in there and do that. Some music, some Pear Deck joining music. Peppier than that. Oh, it's so boring. There we go. Pear Deck joining music. Let's go. Wow, you guys are fast. Yes, Bryce, but most of them are duplicate slides where you're dragging dots. Okay, that's why there's so many slides, because I made copies of slides and had you drag dots around them, okay? So a lot of it is participation. I promise it won't be so bad, okay? Okay, just give me a minute here. Almost all there. Yes. Okay, hold on just a second. Let me turn this off. Oh man, can't get anything to work today. Okay, yes, Julia, what is it?
Connecting. She wanted me to ask if you could send her a new link for Blackboard Collaborate so she could try to get on.
Yeah, I can do that. I also just sent her the Pear Deck code.
Okay.
So either way, she should be able to get in. But yes, I will resend that. Thanks, Julia.
All right. Thank you.
Okay. All right, let's go and get this party started here. All right. So where am I? Here we go. All right, here we go. So this is from, I'm not sure which book you guys have. It should actually be the right chapter for you. This is Introduction to Metabolism, which has a lot about enzymes in it. Okay. So a living cell is a miniature chemical factory where thousands of reactions occur. You don't need to write that down. Here's what you need to write down. Metabolism is the totality of an organism's chemical reactions, mostly within living cells. That's what metabolism is. That's the definition. It's all the chemical reactions happening in your living tissues, whether they be breaking down things or building up things. Both of those types are included. Your entire metabolism is all your chemical reactions, not just your digestion, but also chemical reactions where your cells are building. Okay, so this is important. Energy coupling. A lot of students get confused about this. What exactly does that mean? And what it means is you've got two opposite reactions where one's releasing energy and one's absorbing it. And often they happen really close together in the cell cytosol, and that's on purpose because one is releasing energy, the other one is then using. So for example, you have ATP. When ATP loses a phosphate, it releases energy. See the little red energy there that then goes to help another synthesis absorbing energy reaction, such as putting two amino acids together to make a dipeptide. Energy released from ATP hydrolysis is used in the dehydration synthesis of a dipeptide. You should remember all those terms. So now here's your chance. Okay. Let's see some dots moving. What are you doing? Okay. A little confusion? Hope it, yeah, you can see that. Yes. Okay, so the green dot is energy releasing. So that's the ATP reaction, right? The ATP reaction is releasing the energy. Amino acids coming together should be the red dots. That's where the energy is being absorbed. Right? So green on ATP, that's releasing it, and red on the amino acids. And whoever's not moving around, I know you're not, but that's the answers. Okay, all right. Okay, a metabolic pathway. This is another concept that is more advanced than your freshman year. A pathway begins with a specific molecule and ends with a product. So you've known a chemical reaction as you begin with one thing and then you end with another thing, and that's the end, right? One chemical reaction. Well, it turns out that most of the chemical reactions that are happening in your cells are in a line that's part of a large pathway like this. So where you have chemical A that is turned into B using enzyme 1. And then chemical B is turned into C using enzyme 2. And it goes a long way, even more than this. And you find this might be the construction of important enzyme, or it might be the breakdown of an important food molecule until you get finally to products that your body needs it to be in. Now, using that, pretend that a red dot will denature an enzyme and drag dot to the place in this diagram that would result in A and B being just fine, but C and D not being created. So where would your dot need to go? Just one enzyme denatured that would cause that. Yes, the dots may be a little bit too big. Yes, enzyme 2. Enzyme 2 is the one because, yeah, there you go. Okay, so A and B are fine because A is the starting product. Enzyme 1 has created B, so they're fine. But then C cannot be created without the help of enzyme 2. And therefore, if C is not created, D cannot be there because enzyme 3 will only fit with substrate C. Excellent job. Good, you're getting the hang of it. And this isn't so bad. Okay, catabolic pathways, emphasis on the correct syllable, release energy by breaking down complex molecules into simple. Okay, so catabolic pathways release it, such as cell respiration, breakdown of glucose in the presence of oxygen. So that's catabolic pathways. Breaking things down, releasing energy. Anabolic pathways are the reverse. They consume energy, absorb it, to build bigger molecules from smaller ones, such as photosynthesis or the synthesis of protein from amino acids, right? Okay, on you. Red dots to the catabolic pathways, green dots to the anabolic pathways. Yes. Okay. Haha. Okay. So catabolic pathways are going to be breaking things down. That would be respiration and starch digestion would be red. So respiration and starch digestion are breaking things down. Green dots go to anabolics. Photosynthesis, anything with synthesis usually is going to be anabolic, energy absorbing. You got it. Good job. Okay. So respiration, catabolic, breaking things down, releasing energy. Starch digestion, also catabolic. Protein synthesis and photosynthesis are going to be anabolic. They're going to be absorbing reactions. Okay, good job. Forms of energy. Okay, energy is the capacity to do work or cause change. It exists in various forms. Here's where you get to show me some written work. How many of these can you name? Okay. All right, good. I think this is for what, 7th grade, 8th grade, probably? Go. Good, active, passive. Active and passive aren't really forms of energy. Getting this, there we go. Oh, geez, there it is. Electric, kinetic, and potential. That's good. All right, so we're learning it. Got a lot of examples here, okay? Motion, renewable, kinetic, potential, okay. Okay, forms of energy. Really, we're talking about potential, kinetic. There is light energy, nuclear energy. There is heat energy, mechanical. Yeah, there you go, thermal energy. All right, this is good. I'm glad we're doing this. the kinetic and potential were the big ones I wanted to make sure you got. Heat also is a really important one for biology. ATP is a molecule that's involved with energy, but it's not a form of energy in itself. Okay, yeah, all right. Chemical is a form of energy, yes. And again, these are molecules here, which can store energy, but they are not forms of energy. So try not to confuse molecules with forms of energy, okay? All right, that's good. That's good, all right. Let's keep going. Let's keep going, all right. There, so these are the main ones. I want you to take a minute to write these down. I'm gonna try to help you out with her link issue here. Hold on a minute. I'll be right back. Just write those down for me for a minute. Here I'm back. All right, kinetic energy. An example of that is heat. We'll talk a lot about heat. Potential energy, and the main example is chemical energy, which is the energy stored in chemical bonds of molecules. And probably the sentence at the bottom may be the most important thing there, that energy can be converted from one form to another. And also that some forms of energy are more useful than others. In terms of doing cell work, heat energy is way at the bottom of the list. So is light. There are very, very few things in cells that can actually use light. The chlorophyll molecule is one. That's it. It can actually use light. Nothing else can. What you want in terms of forms of energy in the cell is the chemical energy within the bonds, particularly ATP molecules when they break. That releases a form of energy that can immediately be used by chemical reactions. Okay, let's move on. Presentation, go. Right. So I don't know. I used to think about this when I was a kid a lot because I'm a total geek. When you climb stairs in your house, you are raising your potential energy. When you fall down those stairs, you are experiencing kinetic energy. That's basically it. But in terms of this, we're really thinking about molecules here. Okay, laws of thermodynamics. There are two of them. I don't need you to write any of this down. I just want to read it to you really quickly. Don't bother writing this. Thermodynamics is the study of energy transformations. An isolated system, like a liquid in a thermos, is isolated from its surrounding, right? But in an open system, energy and matter can be transferred between the system and its surroundings. Organisms are open systems. So is the Earth, by the way. We receive light energy and energy, heat energy from the sun, and then we radiate. The Earth radiates back into space. A lot of that heat energy gets lost forever. We can't get it, which is why sunlight winds up being so important. Okay. Here you should write this down. The first law of thermodynamics, which you probably learned in your chemistry class, matter cannot be created or destroyed, or the conservation of matter and energy principle. All those stoichiometry problems that you may remember doing, right? Where you had to have the same number of moles of things on one side as there were on the other. Or if you had six carbons on one side, there needed to be 6 on the other, and it all had to be perfectly balanced. That's the first law of thermodynamics. The second law of thermodynamics is during every energy transfer or transformation, some energy is often lost as heat. Every energy transfer or transformation increases the entropy or disorder of the universe. You don't know that word yet. I know. We're going to get to it. Okay, was that too fast? Do I need to go back? Okay. I'm just going to go on then, okay? Okay, so here's up to you. Which of the following correctly describes the first law of thermodynamics? There we go. Okay. So the first law is about conservation of energy and matter, right? So that would be B. Energy and matter can be created or destroyed is the right answer here. A and C are the second laws. We'll get to in a second. So good for you if you got that out of the bat. The next one. Okay, so now, which of these correctly describes the second law of thermodynamics? Okay. Getting there, getting there. Right. A little energy always lost as heat. Yes. So C is the first law. Anything where something's being conserved or things can be transformed but are always conserved, that's the first law of thermodynamics. The second law talks about how with every energy transfer, there's a little bit of energy lost as heat. And there's also a new word in there called entropy that we're going to talk about next. Okay. A is the right answer here. The second law of thermodynamics says that with every chemical reaction, a little energy is always lost as heat. With every energy transformation. Okay. This. And I just like this bear picture. So here's the first law example is that chemical energy is going to go into the bear, and then the bear is going to, incorporate some of that into its own cells, and then some of that's going to be used that way, and then some of it will be lost as heat, which is conservation. Right? The second law of thermodynamics is that with all the metabolic reactions happening inside this bear, all those transfers between the chemical energy in the cells of the fish versus what gets incorporated into his muscles and energy use, all those transformations have energy lost as heat happening from them. So there's the second law. Okay. The second law mentions entropy. New word, right? What is entropy? Entropy is a measure of disorder or chaos. So I'm sure some of you at home have room for this. This is entropy. This is chaos. This takes no energy to get your room to look like this, does it? It's a downhill situation. At least it always was for me. If I don't tend to my room, it's gonna look like this. But going the other way and creating order from disorder takes energy, doesn't it? Right? An uphill. That's an uphill climb. Getting your room back to looking neat takes energy input. So just think about that while we're going through this. Okay. Drag the thought to the image showing the greatest entropy. Easy one, guys. right? Which one of these rooms is showing the greatest entropy? Entropy, which is otherwise known as chaos. Yes, you got that one. Good for you. Okay, all right. All right, so definitely. Heat, we got to talk about heat. Heat is a form of entropy. It fits with the second law of thermodynamics. Every chemical reaction loses a little energy as heat, thus increasing the entropy of the universe, right? Because some reactions do decrease the over entropy of the universe if their products are more organized than their reactants. Okay, so now we're going to talk about disorder and order in terms of molecules and in terms of numbers of molecules and size of molecules on two sides of the reaction. So let's look at that. Drag the dot to the image that shows the greatest entropy now. This is a glucose molecule over here. This is a bunch of carbon dioxides and water molecules over here on the right. Which one of them is less ordered, less organized, more chaos? Exactly right. Yes, just like the messy bedroom, a whole bunch of little carbon dioxides and waters is much less organized. It has more entropy than the organized, put together, connected glucose molecule. Excellent. All right, keep that in mind as we go on to the next ones. Okay. Living cells convert organized forms of energy into heat. This happens all the time. And these are called spontaneous processes, or you could also call it kind of a downhill process. You don't need a lot of energy to make a lot of entropy. Just like it doesn't take much energy to just allow your room to get messy, right? So those are called spontaneous processes, usually where you're starting with a few really organized molecules and breaking them down into lots of little molecules. Okay? Order and disorder. Living things are constantly fighting against your own entropy. Our cells synthesize large ordered molecules from thousands of tiny ones. Protein synthesis takes hundreds of amino acids with lots of entropy, right? And builds them into one large protein. Does this reaction increase entropy or decrease it? Explain your answer. Here's another place to write some answers down. Go ahead and do that. Right. Mm-hmm. All right. You're getting the idea. This is good. This is good work, guys. Well, it does decrease it. Yes, decreasing entropy because you're taking lots of amino acids and organizing it into one single big protein decreases the entropy. Yes. Yes, I like organizing a bunch of little things into one big one. Very nice. It organizes all the scattered amino acids into one unit. Yes. So if we are decreasing entropy, we are very often doing something that is anabolic, right? There's that other word we learned about, where we are probably absorbing energy in order to create that order in the large protein molecule. Decreases it. Very good. You're all getting it right. Excellent job. Okay, next one. All right, same thing as before, only this time, just the red dot to processes that increase entropy of the universe. Increase it, right? So which one of these processes would be increasing chaos? Or maybe taking, oh, I'm just going to be quiet. You tell me. You can do it. Okay. All right. Most of you got it. So respiration is definitely one, because that's taking a glucose molecule and making lots of carbon dioxides and water, right? So that's increasing the chaos. STO is starch digestion because starch is a polysaccharide, big molecule, and you're breaking it down into lots of individual glucose molecules, right? There you go. You got it. Excellent job. Okay. So increasing entropy of the universe would be respiration and starch digestion. Decreasing it would be building a glucose molecule from lots of carbon dioxides in water, like photosynthesis, or building a protein, like we just talked about. Okay. This is just some ways in which living things organize molecules and organize the energy that's available to them into these complex, amazing patterns, right? And your body is constantly fighting against its own entropy. That's what we're doing. Life means that you are using energy to stay organized while all through your body your body is constantly trying to become disorganized. All the digestion of food and all of the breakdown of toxins and all of the breakdown of your body, you're constantly fighting against that, repairing tears and fixing things. And it's once that when your body dies, that entropy finally takes over. And then all of the entropy continues until you have completely decayed. You can think of life as a metabolic fight against the powers of entropy. The free energy change of a reaction. Okay, so again, don't write this down, just let me read it to you. Okay? I just want to know which reactions occur spontaneously and which require the input of energy. So you need to determine energy changes that occur in chemical reactions, and you can graph them. So first of all, we need to have some variables. These are good variables to write down. I'm going to give you time for this screen, okay? Delta G is called the free energy change. And that's the difference, and it can be a calculated difference, between the amount of potential energy of reactants and the amount of potential energy stored in products. Delta S is the symbol for energy. E sub A is activation energy of a reaction, or the amount of input of energy needed to get a reaction started. So those are the three very important symbols you'll need to really know how and where to put them on the graph that you're about to see. Yeah. Yes. Okay. So this is called a free energy graph. Somebody may have a question? Too fast? Oh, okay. I'm going to go back. I'm sorry. That's what you to tell me. I'll go back. Sorry. Here we go. Okay, I'm just going to leave it here for a minute. Take your time. It's okay. I'll be right back. Okay, are we ready to go on? Is that better? Yes? Okay, here we go. Okay, here we go. Okay, here's a question page. That delta G, Which one of these letters do you think it might be, according to your definition of delta G, of the free energy change? Okay. Working out. There. Okay, so I believe, hold on, let me go back to, yeah. It's C. C is the one, okay? So this right here, C is the delta G. And the delta G, remember, is the difference between the products, energy stored in the products, and the energy stored in the reactants. So your X value here is time. This is just something that's happening over time, the time lapse of a chemical reaction or a chemical change. And the free energy is usually measured in something like kilojoules or something like that. And it's the amount of energy stored in the reactants, in the covalent bonds of the reactant molecules is a set value right here. And then that amount of energy stored in the products is a set value here. And you can calculate the delta, right? The delta G, which is what C is the amount of free energy change. A and B are actually what? What would they be? What do you think? Looks like there's a little bump before the reaction really gets started. So what would you think that would be? A and B are actually the same thing. It's just a matter of whether the enzyme is there or not. Any idea? Yes. Tell me.
Activation energy.
Beautiful. Yes. You remember your ninth bio. Good for you, Mikhail. Excellent. Yes. Good. All right. Okay, so let's keep going. Right. Okay, so this graph is a fully labeled free energy graph, and you have a picture on your worksheet that I gave you. So if you wanted to fill in what those things mean now, you can do that. And I'm just going to leave this up for a minute. Okay. If you don't have the worksheet printed out, you can just sketch this on a piece of paper. It's pretty easy. Just make a curve, a couple of bumps there, and you know, you guys are artistically talented enough to handle that, I think. And if not, you can come back and get it later because I'm going to post this on your Google Classroom. So don't ever feel like, you know, you can't go back and get it later. And I'm recording all these classes where I'm doing a lot of teaching. I'm always recording these. So you can also go back and find those. So there's your delta G. The larger activation energy is the one that's without and uncatalyzed, means there's no enzyme present, and means there's more activation energy. It takes a lot more energy to get this reaction started. When you add the enzyme, the activation energy is a lot less. And then one important thing to realize about this picture is that the delta G doesn't change regardless of whether you have a big activation energy or a small one. So adding the enzyme doesn't change the overall change in energy of the reaction, but it'll change how fast the reaction can happen or whether it can happen at all. I'm going to keep going. Okay, so these are some words also that are on your worksheet that I think are good contrasting words. We did anabolic versus catabolic, right? And now exergonic versus endergonic are very similar to catabolic and anabolic. An exergonic reaction is a reaction that releases energy. So very often that goes along with catabolic. And an endergonic reaction absorbs energy. And that often goes along with anabolic. So there's some new vocabulary for you. Okay, yes. here we go. All right, so here's a question. Is this curve exergonic or endergonic? What do you think? Do you think this reaction is releasing energy into the environment or absorbing it? Mm-hmm. Okay, let's see what you said. It is exergonic, yes, because the reactants have more potential energy than the products. So that energy must have left, right? So if the reactants reacted and changed into the products, that energy that used to be inside their chemical bonds got released somehow. Good. Nice job. Okay. All right. Now this is another one. You have two contrasting graphs on your worksheet, and this is the one I can tell you what to put on those graphs. So the one on the left, and I think it's the same as the one I put on your worksheet, is endergonic. So the reactants are lower, have less potentially energy in them than the products. So an endergonic reaction is going to absorb energy, right? Whereas an exergonic reaction over here is going to have its reactants have more potential energy in them than the products do, and so it will release it. And then the other things that are good to write down is the delta G, you notice, is the same in both. But this delta G in the endergonic reaction is going to be a positive value. This delta G is positive because it's being absorbed into the products. This delta G is negative. Negative delta Gs are going to be released, that energy is released into the environment, either as heat or possibly, if it's coupled with another reaction, it might go into chemical bonds of other molecules. Okay. Here's a question. Same graphs. Drag the red dot to the graph that could be showing respiration. The green dot to the one that's showing photosynthesis. And then after this, I think we need to take a break. I know it's been longer than I thought it would. I apologize. Take a look at that. Okay, how are you doing? Okay, most of you, yes. So the photosynthesis is endergonic. That's going to be absorbing energy, the energy of light, right? So green is the photosynthesis one. That's over here on endergonic. And red is respiration. That's exergonic. That's energy releasing. Excellent job. Okay. All right. You guys need to take a break. I'm sorry. It took a lot longer to get through that much than I thought. So I want you to just go and stretch. You've gotten most of what I wanted you to get. We're going to go over enzymes just a little bit when you get back. And we might even have time to work after that. So just take a break right now. Just go take a break. I'm going to shut down and play some music, okay? Go and dance, go put your feet in the grass. As long as the music is still playing, it's still break time, okay? Break time. Go away from your screen. Okay, there we are back again. All right. All right, here we go. Let's go back into this. Let's see how you did on here. Right. Yes. Okay, good. I think we already did that, right? Yes. Right, moving on. All right, so now this is the enzymes part. How do enzymes fit into all of this? Hopefully this will be kind of a review of 9th grade. This shouldn't be too difficult for you. There's a few extra little things you might not know about enzymes, but I bet a lot of this is review. So an enzyme is a protein, a catalytic protein that speeds up a chemical reaction without being consumed by it. Activation energy. You don't have to write this down. I just put it in here as something to read to you. Don't write this down. Every chemical reaction between molecules involves bond breaking and forming. This initial energy needed to start a chemical reaction is called activation energy, and it's the bump on the front of the curve. It's often supplied in form of thermal energy that increases molecules moving around, and that makes them more likely to bump into each other the right way. But usually that's not enough to make a reaction go fast enough to supply a very greedy organism in terms of energy like a human being. Okay, how enzymes lower this barrier? Enzymes do not affect the change in free energy, right? Because that delta G never changes on the graph. But they can lower the activation energy, the bump in the beginning. And this is the way they do it. You don't write this down. Here we go. So I think you already have this. But what they do is they lower this curve right here. They lower this. They don't affect the delta G. They just make it faster for the reactants to react and become products by lowering this hump here. Okay, here we go. This I would write down. So I'm going to leave this up for a few minutes. And you really should know this already. You should know that enzymes have specific substrates, right? Enzymes have an active site, which is a region on their structure that binds with the substrate. When the substrate is bound to the enzyme, this is called an enzyme substrate complex. There is something called induced fit, and all that really means is that an enzyme kind of changes shape to fit around its substrate. Kind of gives it a hug. And you should know that each enzyme can only work on one specific substrate. They're very picky. So that's called substrate specificity. I'm just going to let you have this screen for a minute. Let's keep going. Okay, can you give me a thumbs up when you're ready to move on? Excellent. Thank you for that. Okay, and remember, you can always go back and get it later, miss a word or two. Paraphrasing is a wise skill to develop also. Remember that. Okay. All right, so this is a nice little picture here. The blue thing is the enzyme, and the red thing is the substrate. And look at the pocket that's often present on many enzymes that is perfectly the same shape as the substrate. And you see how the enzyme kind of wraps itself around it, gives it a hug. That's called induced fit. That's what induced fit means, is the enzyme kind of temporarily changes shape while it's in contact with the substrate. And that area in there is a microhabitat kind of place that is a perfect place to tug at the bonds and to cause the substrate to change or to break, or maybe to bond to another substrate. And this is part of the reason why enzymes can speed up reactions, is because of the chemical characteristics inside the active site that tug at bonds and provide a perfect place for a chemical reaction to happen. Okay, so here it is. These are the ways that enzymes speed up chemical reactions. Right here, these 4 things. That the active site can orient substrates correctly. If you're synthesizing a sugar, right? If you're synthesizing a sugar from 2 small sugars or protein from 2 amino acids, the enzyme will fit both of those building blocks together right in the right places for their bonds to form. Or if you're trying to break something, the active site will tug on the molecule and cause it to break apart. So it provides a favorable microenvironment, and it can also temporarily bond to the substrate and help to pull it apart or rearrange it. Okay, now you. Write down what you remember, guys, from that screen I just did. What kinds of things are happening in that active site that can help tug a molecule apart or put it together? Or just tell me everything you remember about an active site, because you'll get the answers right, but just by telling me that. What is an active site? What should it be able to do? What kind of characteristics might it have that that would help a molecule to either get pulled apart or put together with another one. Okay, that is a correct sentence. Okay. Oh, Jesus. Oh, I'm so sorry. Technology. Okay, so enzymes can move on, create favorable microenvironments. Yes. It's on the enzyme where substrates go to start chemical reactions. Very nice. They're good orientation of substrate molecules. Yes. Okay. Yes. Oh, I love that. Beautiful. Very nice. Very nice list. You guys must be writing stuff down. That's good. Like that, it causes substrates to fit together better, right? Positioning substrates in the proper orientation. That's lovely. Create an ideal microenvironment. Right. Orienting substrates correctly. Very nice. Yes. That's very nicely written too there. That's beautiful. But you guys are thinking very well. The substrate's position, for example, if you're synthesizing something and putting two new things together, the enzyme will have an active site that fits those two things and orients them better so that new bonds can be formed much more quickly and easily. Yes. Much better than if they were just bouncing around randomly in space and working together that way. Without the enzyme there to orient them and to guide their reaction, a lot of those synthesis and digestion reactions wouldn't happen fast enough for our greedy, big, complicated bodies to survive. All right, excellent job. That's really good. Okay, oops. I did that one. All right, here we go. Now this is probably a series of pictures that you remember from freshman year, right? So if we start here, we have the purple enzyme and we have two substrates coming in. So this is a synthesis reaction. And you see how it orients them perfectly so that the new bond can form between them much more easily. This picture really shows that well. And then look, there, the chemical change happens, bound, and then they're changed and released. In this case, it looks like some kind of a chemical change happened and they didn't get connected. But then the enzyme goes back, reverts to its original shape. So remember, the enzyme is not consumed in the reactions and it's ready to accept more substrates at its active site. Okay, environmental factors. I think we're going to go to that next week. I think we're going to stop right here, all right? So congratulations, you did your pear deck. As long as I can find your name in here, we're good. Don't log out of this. Stay in this, all right? Just stay in it so I can find you and mark you down as having completed it, 10 out of 10, right? I'm going to leave this as is, and we're going to maybe talk about the rest of it, environmental factors, next week, because that really fits in with the lactase lab better. And I think you guys have done enough today. It's kind of overwhelming. Okay, all right. So I'm just going to put a blank worksheet here. There we go. So just stay in the pear deck, but you can go on to do other things now. So now what you should be able to do. Let me just share my camera with you. Okay, so now you can see nice and big on your screen. You're gonna do this, okay? This is the other sort of must-do for today. There's the first page of it, and here's the second page of it. Okay. I think the best way to do this is to print it out and do it by hand. I don't, unless you have drawing capabilities, and you might be able to type things, but I think it will be easier if you print it out and do it by hand and then give me a photo shot or something. But if you can figure out how to do it just on the screen and then turn it in that way, that's okay too, okay? There's always lots of options here. So, so I'm going to put you into breakout rooms now, and you can use the time however you would like. The first must-do for today was the pear deck you just did. So if you were there and I find your name, you're good. The second must-do is this sheet that I just showed you, free energy diagrams question sheet, which is in your Google Classroom right now. and you each have your own copy, okay? And then your homework is to do that HHMI Ed puzzle, which is actually summative and is your pre-lab for next week, which you can get that done again on Monday, okay? And that's it. So I'm going to put you in breakout rooms, and if you have questions for me or other issues, you can come and see me. And if not, just hang out until class is over and get what you can done, because it's always a good thing to do. One room for me. And then let's do. There's a work alone room like that. Okay. All right. How is everybody feeling? I know that was a lot of information, but I think sometimes it's good to directly give you stuff that's harder. You guys good? Can you give me a thumbs up if you're good? Are you there? Yeah. Okay. Okay. Good. Excellent. All right. All right. So breakout rooms now, and as always, use your time well. And when it's almost time to go, I'll call you back in and say goodbye. All right. Here we go. Where's the thing? Start.